So what we are going to do now is the second disaccharide, which is nothing but maltose. Maltose. I have already told you sucrose. Sucrose is a disaccharide, consists of alpha D glucose, and beta D fructose. Okay, actually speaking, alpha D glucopyranose, right? But I'll just use this term, alpha D glucose and beta D fructose. Okay, they link together by means of glycosidic linkage. Okay, between in glucose it is carbon number 1 and in fructose it is carbon number 2. Okay, so between these we have glycosidic linkage, right? C1C2 glycosidic linkage, okay, exists, and this is what we call as sucrose. Okay, sucrose is a disaccharide which is made up of two monosaccharide units, alpha D glucose and beta D fructose, and they are connected through C1 of glucose and C2 of fructose. Okay, and you know very well, C1 is anomeric carbon in glucose, C2 is anomeric carbon in fructose. Since both these anomeric carbon atoms are involved in glycosidic linkage, there is no free hydroxyl groups are present. There is no free hydroxyl group on C1 as well as on C2. Therefore, the ring cannot be opened. So this sucrose is a non-reducing sugar, I told you, because the ring cannot be opened. So free aldehyde group will never form. Therefore, sucrose is a non-reducing sugar, so which we discussed in the last class. And I have also told you about invert sugar and all. Okay, then the second disaccharide that we are going to discuss is maltose. Maltose is a disaccharide, contains alpha D glucose, one monosaccharide, and alpha D glucose, this is another monosaccharide. So two alpha D glucose units, they combine, or they are connected through glycosidic linkage, and here the two glucose units are connected through in one glucose it is C1 carbon, in the other glucose it is C4 carbon. So there is a glycosidic linkage between C1 of one glucose unit and C4 carbon of another glucose unit. So C1 C4 glycosidic linkage exists in the case of maltose. Okay, yeah. Then so what is the structure? The structure is very simple, right? What you have to do, you draw the Haworth projection, right? The Haworth structure for alpha D glucose. So this is carbon number 1. Alpha means you must put OH group down. Alpha means in carbon number 1 you should put OH group down. And in glucose, you know the structure of glucose. Second carbon OH group down, third carbon OH group should be up, fourth carbon down, fifth carbon, I have told you, always hydrogen down, CH2OH up. Okay, and you know very well, this is the numbering. This is carbon number 1, this is carbon number 2, 3, 4, this is carbon number 5, and this is carbon number 6. This CH2OH carbon, no, this carbon is actually carbon number 6. Okay, so this is one alpha D glucose unit. Now I'll draw another one. Let me draw here. Alpha D glucose, so put OH group down. Remaining carbon groups, you should keep it as such. Okay, like this structure, H, OH, here H down, OH up, here OH down, H up, here always H below, CH2OH above. Okay, so you take two alpha D glucose. Now what I'm going to do, this is carbon number 4 of this glucose unit, and this is carbon number 1 of this glucose unit. Now you combine the two OH group and remove one water molecule. If you remove one water molecule, what will I get? I'll just remove this OH group. If you remove one water molecule, then one oxygen will remain. That oxygen will form a link like this. Okay, between C1 and C4 you will get a link, and this is what we call as 1,4 glycosidic linkage. Since it is alpha, okay, with respect to this glucose, okay, this OH group is already present in alpha position, so it is called alpha, and this is carbon number 1, alpha 1, and this is carbon number 4, so 1, 4, alpha 1, 4 glycosidic linkage. Glycosidic linkage exists, okay, between carbon 1 and carbon 4 in maltose. This is the structure of maltose. Okay, I just removed these two OH groups. I have eliminated a water molecule and I connected the two carbon through an oxygen atom. So this is basically ether linkage. I have told you already, it is basically an ether linkage, and this ether linkage exists between two monosaccharides. If ether linkage exists between two monosaccharides, we call that linkage as glycosidic linkage. Okay, so keep this in mind. This is maltose. Students, observe this carefully. It's a very important point. In maltose, how many OH groups are there? Can you count and tell me number of OH groups in maltose? Yeah, so there are, okay, 1, 2, 3, here 4, similarly here we have 4, okay, 5, 6, 7, 8. So there are 8 OH groups. 8 OH groups are present, right, in maltose. And another important one here, listen, in one glucose unit, C1 carbon, actually C1 carbon is the anomeric carbon. In C1 carbon there is no OH group now, okay, it become ether. It is involved in ether linkage or glycosidic linkage. So in C1 carbon there is no OH group in one glucose unit. In the second glucose unit, see, this is carbon number 1 and we have OH group. Students, observe carefully. Whenever anomeric carbon contain OH group, then ring opening is possible. So here this carbon in glucose, C1 carbon is the anomeric carbon. In this glucose unit, C1 carbon does not contain OH group, whereas in this carbon, in this glucose unit, C1 carbon has OH group. Therefore, if you put this compound in basic medium, the ring get open up and you will get a free aldehyde group. Free aldehyde group will come in the open structure, okay, and that aldehyde group, okay, reduces tolerance reagent, right? Therefore it is a reducing sugar, okay, it is a reducing sugar. It's a reducing sugar, no doubt. Maltose is a reducing sugar. Why? Because in one glucose unit the anomeric carbon, that is C1 carbon, contain a OH group. Therefore ring opening is possible. In sucrose it is not possible. Why? In sucrose the anomeric carbon of glucose and the anomeric carbon of fructose, that is C1 and C2. C1 is anomeric carbon in glucose, C2 is anomeric carbon in fructose. Both are involved in glycosidic linkage. Therefore there is no OH group in C1 as well as in C2. C1 of glucose and C2 of fructose, there is no OH group. Therefore ring opening is not possible here. Therefore this is a non-reducing sugar, whereas maltose is a reducing sugar. Right? Keep this information in mind, very, very important. And what's the linkage? The linkage is alpha linkage between carbon 1 and carbon 4. That's why I wrote like this, alpha 1, 4 glycosidic linkage. Okay, then the next sugar that we are going to discuss is lactose. Right, students, any doubt in this? Any doubt?
No, sir.
Yeah, right. Then next sugar is lactose. Lactose is also a disaccharide. Okay. In lactose consists of two monosaccharide units. What are they? Beta D galactose and beta D glucose. See this, very important. Both are beta D. Beta D galactose and beta D glucose. Okay, they are connected through glycosidic linkage and form lactose. Right. So remember, if I hydrolyze lactose, you can say in this way also. You take lactose and hydrolyze it. Hydrolysis. If I hydrolyze lactose, I will get galactose and glucose. I will get beta D galactose plus beta D glucose. Okay, that's the meaning. If you hydrolyze it, the glycosidic linkage will break and you will get the two monosaccharide units. Okay, so lactose is a disaccharide. Why? Because one molecule of lactose will produce two monosaccharide units on hydrolysis. Okay, now, and you also know that this information I have given already. Glucose and galactose are C4 epimers. Students, remember this. Glucose and galactose are C4 epimers. They are epimers. C4 epimers. What is meant by C4 epimers? What's the meaning? The meaning is, listen, let me draw the open chain structure so that you can easily understand this. Open chain structure, right? So first I will draw the structure of glucose. 1, 2, 3, 4 chiral carbon atoms are there in glucose. And you have an aldehyde group here, aldehyde. This is carbon number 1. Carbon number 2, OH group right hand side. Carbon number 3, OH group should be in the left hand side in Fischer projection. 4 and 5 always OH group in the right hand side. OH group should be in right hand side in glucose. Okay, this is the structure of glucose. This is D glucose. Listen, this is carbon number 2, this is carbon number 3, and this is carbon number 4. Now what I am going to do, I am going to draw the structure of galactose by changing the position of OH and hydrogen in carbon number 4. CHO 2, 3, 4, 5, CH2OH. Okay, I'll keep all these groups as such, right? In carbon number 2, in carbon number 3, okay, in carbon number 5. What I'm going to do, I'm going to change the I'm going to rearrange, okay, the OH group and hydrogen of carbon number 4, right? So what I'm going to do, I'll put OH group in the left hand side and hydrogen in the right hand side. This is what we call as D-galactose. What are epimers? Epimers are, listen, epimers are they are isomers, right? They are basically isomers. They differ, the two isomers differ only in one chiral carbon. Okay, see this fourth carbon is a chiral carbon. Here also the fourth carbon is chiral carbon. What is the difference? Okay, in Fischer projection, right, the fourth carbon of glucose contain OH group in the right hand side, whereas in Fischer projection the fourth carbon of galactose contain OH group in the left hand side. All other things are same. All other things are same. That's why they are called epimers. So remember, glucose and galactose are C4 epimers, right? And I have also told you glucose and mannose. Glucose and mannose are C2 epimers. Okay, we have already discussed about that. Okay, so you just go through our notes. Now what I'm going to do is that I'm going to draw the structure of lactose. Okay, for which first you should draw beta D galactose and beta D glucose, then we have to connect. Okay, yes, how to proceed. Right, so first let me draw glucose unit here. This is alpha beta D glucose. I'll draw beta D glucose in the right hand side. Okay, beta means you should place OH group up. Students, observe carefully. In carbon number 1, if you place OH group up, it is beta. Okay, I am going to draw the structure of glucose. So second carbon OH group down, third carbon OH group up, fourth carbon OH group down, fifth carbon always hydrogen down, CH2OH up. Okay, so in this is carbon number 1 and this is carbon number 2, 3 and this one is carbon number 4. This is beta D glucose. Okay, now I'll draw beta D galactose. I have told you glucose and galactose are C4 epimers. So I'm going to keep this beta D structure as such. Okay, so this is beta D galactose. Okay, so since it is beta, we should keep OH group up in carbon number 1. This is carbon number 1, hydrogen down. Okay, second carbon you keep it as such, second carbon you keep it as such, third carbon you keep it as such, but don't forget in fourth carbon of galactose, okay, hydrogen should be down, OH should be above, because glucose and galactose are C4 epimers. Okay, C4 epimers. So remember this. Then fifth carbon always hydrogen down, CH2OH above. Right? Now what I'm going to do, I'm going to form the glycosidic linkage between carbon 1 and carbon 4. How? Listen, now this OH group is above and this OH group is below. So I'm going to remove a water molecule between these two and I'm going to form an ether linkage. Okay, let me draw here. Just remove one water molecule from this and you'll get a structure like this. This is how you must draw the structure. Okay, so here we have OH group above, hydrogen below. Here hydrogen above, OH group below. Here OH group above, hydrogen below. Then fifth carbon hydrogen below, CH2OH above. Okay, here let me just, I'll keep this bonds as such, we'll connect it. Okay, and here you have to keep the group as such, H, OH, OH, H, H, OH, H, CH2OH. Now what I am going to do, I have I'm going to remove water molecule from these two OH group. Okay, if you remove H2O, one oxygen will be remaining. Right, one oxygen will remain, and I am going to connect that oxygen. Using that oxygen, I am going to connect C1 and C4. How? See, this way. Here we have hydrogen, here we have hydrogen. Now we have connected carbon 1 and carbon 4. See, sorry, this is carbon 4 and this is carbon 1. I have, very sorry, this is carbon 1, this is carbon 4. I have connected carbon 1 of galactose with the carbon 4 of glucose through beta glycosidic linkage. Okay, students, observe carefully. In alpha glycoside linkage, okay, we draw in Haworth projection. It's the usual way of drawing the structure. In Haworth projection, the bond will be like this. Okay, it will have this type of shape. If you have bonds like this, right, it is usually called alpha. Okay, whereas if you see bonds like this, okay, if the glycoside linkage exists like this, then it is beta. So here in lactose, between galactose and glucose, we have beta glycosidic linkage. With respect to galactose, it is beta, and carbon 1 of galactose and carbon 4 of glucose are connected. Carbon 4. So it is called beta 1, 4 glycosidic linkage. Beta 1, 4 glycosidic linkage. Okay, this is lactose. Now, once you finish the structure, immediately you have to look upon the anomeric carbon. Students, see this anomeric carbon. Is there any OH group available in galactose unit in carbon 1?
No, sir.
No OH group. Whereas here, carbon 1 has now a OH group, right? So the glucose unit, in glucose unit, carbon 1 has one OH group. Therefore, if you put this compound in tolerance reagent, immediately, or in basic medium, immediately the ring get open. This ring will open. Once the ring is open, you will get free aldehyde group. Therefore, the compound will reduce tolerance reagent. So lactose is also a reducing agent, a reducing sugar. Is also a reducing sugar, right? So among sucrose, maltose and lactose, only sucrose is non-reducing among these three. Okay, other things are reducing, right? Maltose and lactose are reducing sugars. Okay, yeah. So keep all these things in mind. Now let me move on to the next one. So with this we have completed disaccharide. Now we'll start the polysaccharide. Okay, so the next one is polysaccharides. Polysaccharides. We also call this as non-sugars. Non-sugars. Polysaccharides are also called as non-sugars. Okay. Remember, this is very important. If you hydrolyze a polysaccharide, you take some polysaccharide, okay, hydrolyze it. Okay, do complete hydrolysis. Complete hydrolysis means you should break all the glycosidic linkage. Complete hydrolysis means we should break all the glycosidic linkage. Once all glycosidic linkages are broken, then you will get only monosaccharides. If you get more than 10 monosaccharides, this is a definition purpose I am saying, if you get more than 10 monosaccharide units, more than 10 monosaccharides, okay, we call that carbohydrate as polysaccharide. Right? Usually it will give 100,000 monosaccharide like that. Okay, you will get huge number of monosaccharides. Okay, but definition purpose I am saying like this, right? If you get more than 10 monosaccharide units, we call that carbohydrate as polysaccharide. Okay, then in polysaccharide we have actually two types. Okay, homopolysaccharide and heteropolysaccharide. Homopolysaccharides, the other one is heteropolysaccharides. Heteropolysaccharides. Homo means very simple, you take a polysaccharide, carry out complete hydrolysis and analyze the monosaccharide. If all monosaccharides are same, Students, observe carefully. You take a polysaccharide, carry out complete hydrolysis. Once the hydrolysis is over, analyze the monosaccharides. If all monosaccharides are same, right, consists of same monosaccharides, maybe all are maybe made up of glucose, or all are maybe made up of some other monosaccharide units, right? If a polysaccharide is made up of a single type of monomeric units, single type means it consists of only glucose units, or only a galactose unit, or only arabinose unit, right? Whatever maybe it is, we call those polysaccharides as homopolysaccharides. Then what is hetero? Students, can turn on your camera. Students, turn on your camera. Hello? Hello? Students, am I audible? Turn on your camera. Right. Then heteropolysaccharides. Okay, what are heteropolysaccharides? Heteropolysaccharides are polysaccharide. They consist of different monosaccharide units. Glucose, galactose, mixtures, right? They consist of mixture of monosaccharides. Let me use the word mixture. Okay, it consists of actually different types of monosaccharide units. I'm just for understanding purpose, I'm using the term mixture. You have a mixture of monosaccharide units. Right, mixture of monosaccharides. Right, that means different types of monosaccharides. Glucose, galactose, arabinose. Okay, if you have a mixture of monosaccharide units, we call those polysaccharides as heteropolysaccharides. If you have same monosaccharide units, it is called homopolysaccharide. Okay, so in polysaccharide we have two types, homo and hetero. Right, in we are going to focus only on homopolysaccharides. Homopolysaccharides. Okay, as I told you before, homopolysaccharide means if you hydrolyze this polysaccharide, you will get only one type of monomeric unit. Okay, in homopolysaccharide we have lot of examples. Okay, starch, cellulose, glycogen, right, etc., etc. Many examples are there. These are all examples for homopolysaccharides. Okay, what is the monomeric unit present? In starch, the monomeric unit present is, starch is made up of many number of glucose unit. Glucose is the monomeric unit present. Okay, in cellulose, glucose is the monomeric unit present. In glycogen also, glucose is the monomeric unit present. Right, so here I have taken few polysaccharide, homopolysaccharide. These are all made up of glucose units. Okay, many glucose units connected through glycosidic linkage and form starch, cellulose, glycogen, etc. Okay, now we will discuss about starch. Okay, starch is a homopolysaccharide. Let me write in the next page. Yeah, students, are you able to follow? Any doubt in this?
Sir, can I show the previous page for a minute?
Pardon?
The previous page, sir.
Previous page, yeah.
Yes, sir. Thank you.
Right. Now, so in homopolysaccharide we have starch, cellulose, glycogen, etc. First we'll take starch. Okay, starch is a homopolysaccharide. Okay, actually speaking, starch is a mixture. Starch is a mixture of, okay, it consists of two fractions. Okay, starch is a mixture of amylose and amylopectin. Pectin. Starch is a mixture of amylose and amylopectin. Students, observe carefully. Starch is not a pure substance. It is actually a mixture. It consists of amylose and amylopectin. Both are homopolysaccharides. Okay, right? Amylose is a homopolysaccharide. Amylose is a homopolysaccharide, and the very important point is amylose is linear chain polymer. It's a linear chain polysaccharide. It's a linear chain polysaccharide. Very, very important. Amylose is linear chain polysaccharide. Okay, and amylose is made up of many glucose units. Right, many glucose units. They are connected through glycosidic linkage, and we'll get the linear chain polysaccharide. Now, how they are connected? How the glucose units are connected in amylose? Students, this is very important point. Keep this in mind. In amylose, glucose units, adjacent glucose units are connected through alpha 1,4 glycosidic linkage. Alpha 1,4 glycosidic linkage only. They are connected through alpha 1,4 glycosidic linkage only. Why I am using the term only, that we'll discuss. Okay. Now, students, I am talking about amylose. Starch is actually a mixture. It consists of amylose and amylopectin. Okay. Amylose is a linear chain polysaccharide where we have alpha 1,4 glycosidic linkage between adjacent glucose units. So let me just show you one or two units. It is actually a linear chain polymer. Okay, it's going like that. Okay, you can take many units. Now they are connected. See, carbon 1 and carbon 4. This is carbon 1 and this is carbon number 4. Carbon 1 and carbon 4 are connected through glycosidic linkage. Alpha glycosidic. Alpha means the bond should be like this. I have told you already. If the bonds, if the ether linkage, if you draw like this, it is alpha, and put hydrogen here and hydrogen here. Okay. Similarly, here also this group will go. It goes like this. Many units, right, will come. Similarly, here between these two carbon, this is C1 of this glucose unit and this is C4 of this glucose unit. So connect the two through alpha glycosidic linkage. Alpha 1,4 glycosidic linkage. Like that it goes. Okay, like that many glucose units are connected. Remaining groups, okay, you just keep it as such. I didn't show that. In one glucose unit I'll show second carbon OH group down, third carbon OH group up, and fifth carbon always hydrogen down, CH2OH above. Okay, same thing should be placed, same groups, right, same arrangement should be there in the other units also. I didn't show that, but this is how you have to draw. In amylose we have only 1,4 glycosidic linkage. Alpha 1,4 glycosidic linkage alone exists. Therefore this polysaccharide will appear like a linear chain polymer. Okay, but linear means don't think that it is straight chain. It's not like a straight line. Okay, the polymer is linear, but in solution it will appear like helical structure. It will appear like helix like this. Okay, it is a linear chain polymer, but in solution, right, they usually appear like this. Okay, so it has a coil like structure, right? And students, this is very important. If you put iodine in starch, you will get blue color, right? You will get deep blue color. Why? If I put iodine, these iodine molecules get trapped in this coil, right? In these coils of amylose, and you'll get a compound, a complex called inclusion complex. That is the reason why we get blue color. So let me draw here. So what I'm trying to say is, you'll get in this coil-like structure, iodine molecules get trapped. Here I will take an iodine molecule. This iodine molecule is trapped in this coil. Just for understanding purpose, I'm showing like this. Like this, in each and every coil, many iodine molecules get trapped, and we'll get a complex called inclusion complex. It is called inclusion complex. This inclusion complex is formed when we add iodine in starch. When we add iodine in starch, the amylose combined with the iodine form an inclusion complex. Same complex is also formed in amylopectin. Okay, same complex is also formed in amylopectin. The mixture of these two, amylose with iodine, amylopectin with iodine, will give you the deep blue color. Okay, right, and this is called inclusion complex. Keep this in mind. Okay, this is regarding amylose. So starch is a mixture of amylose and amylopectin. We have discussed amylose here. And remember, student, starch is insoluble in water because it is a non-sugar. I told you, it's a non-sugar. It is insoluble in water, but in hot water it is soluble. Okay, not very much soluble. To some extent it is soluble. That's why in your laboratory they will provide starch in solution form. Okay, because in hot water, right, it is soluble in some small extent. Okay, then, so this is what is called amylose. Now let me draw amylopectin. Is this clear, student? Any doubt in this? Any doubt?
No, sir.
Okay, right. Now let me draw the next one. Students, turn on your camera, everyone. Turn on your camera. Right. Now, the second one, that is amylopectin. Amylopectin is another fraction present in starch. Okay, amylopectin is a homopolysaccharide, no doubt in that. This is also a homopolysaccharide. Amylopectin is a homopolysaccharide. This is present in starch. It is a homopolysaccharide. Okay, and in amylopectin we have two types of connections between glucose units. Two types of connections. What are they? Number one, we have alpha 1,4 glycosidic linkage, as usual, glycosidic linkage. Not only that, we also have alpha 1,6 glycosidic linkage. Both are present. Very, very important. In amylose, only alpha 1,4 linkage, but in amylopectin we have two different types of linkages, and because of these two different types, okay, the structure is not linear. Okay, actually amylopectin is a homopolysaccharide, but it is not linear, it is branched polysaccharide. It's a branched polysaccharide. It is not linear chain, it is branched. It has branching. How? Let me show this here. Listen, let me just draw a few molecules. 1, 2, 3. Between this we have alpha 1,4 glycosidic linkage. Between this we have alpha 1,4 glycosidic linkage. Okay, so this is going like that. Okay, and similarly, suppose I have a linkage here. Let me draw like this. So here I have another chain containing alpha glycosidic linkage, right? The chain goes like this. Now this is carbon 1. Listen, carbon 1, and in this glucose unit this is carbon number 6. Here we have hydrogen, this is carbon number 6. Between this carbon and this carbon, actually we have CH2OH here, and here we have OH. The two OH group, just eliminate a water molecule and connect them through oxygen. So this is, with respect to this unit, this is alpha, because OH group is below alpha. Now we have an ether linkage between this monosaccharide and this monosaccharide, but this is carbon number 1 and this is carbon number 6 of this unit. Okay, therefore this linkage is called alpha 1,6 glycosidic linkage. Let me just show you here. So this is alpha 1,6 linkage, whereas these are alpha 1,4 glycosidic linkage. So in amylopectin we have linear chains, and between them branching also exists. Between them there are branching. Therefore amylopectin is a branched chain polysaccharide or branched polysaccharide. Amylopectin is a branched polysaccharide. Okay, we have two types of linkages, alpha 1,4 and alpha 1,6 glycosidic linkage. Is this clear? Students, any doubt in this? Is this clear?
Yes, sir.
Hello, students, am I audible?
Yes, sir.
Right.
Yes, sir.
Yeah. So remember, starch is a mixture. It consists of two fractions, amylose and amylopectin. Amylose is a linear chain polysaccharide. Amylopectin is a branched polysaccharide. Okay, in amylose we have only alpha 1,4 linkage, whereas in amylopectin we have alpha 1,4 and alpha 1,6 linkages. Okay, now we'll go to the next polysaccharide, that is cellulose. The next polysaccharide that we are going to discuss is cellulose. And you know very well, cellulose is the most abundant organic compound, okay, in the world. Is the most abundant organic compound. Most abundant organic compound in the earth. It's the most abundant organic compound. Okay, and cellulose is a homopolysaccharide. Cellulose is a homopolysaccharide. So homopolysaccharide, and this is also a linear chain polysaccharide. It's a linear chain polysaccharide. Okay, and here what is the monosaccharide unit present? The monosaccharide unit present here is the glucose unit. It consists of many glucose units. So glucose unit is the monosaccharide unit present in the cellulose. Okay, but what is the connection between adjacent glucose unit? Right, glucose unit, adjacent glucose units are connected through beta 1,4 linkage. Beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage exists between adjacent molecule. They consist of beta 1,4 glycosidic linkage. Okay, just let me draw and show the structure. See, I'll take one molecule here. This is one unit, and show the second unit here. And I'll show another unit here. Like this, it actually goes. Okay, it goes like this, and similarly here it goes like this. Right, now what I'm going to do, I'm going to connect. This is carbon 1, this is carbon number 4. I'm going to connect. Here OH group is above. So students, keep this in mind. Hydrogen is below, OH group above. OH group above means it is beta. Okay, in carbon number 4, OH group is below. So this OH group and this OH group, okay, just remove a water molecule from that and connect them using oxygen atom. So this is called glycosidic linkage. So this is 1,4 glycoside linkage, but this is beta with respect to this unit. Okay, because OH group is above. Similarly here, right, we have beta linkage between this carbon and this carbon. We have beta linkage. Right, so this goes like this, right? So you'll get a linear chain polymer. So this is beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage. Okay, take this. Beta 1,4 glycosidic linkage. So most plant body, right, that bark, leaf, right, consists of cellulose, right, and in cellulose the glucose units are connected through beta 1,4 glycosidic linkage. And keep this in mind, students. Human beings have lack of few enzymes. Because of that, we cannot digest, or we cannot hydrolyze, or we cannot break these linkage units. That's why we should not take the vegetables directly. We should cook, we should hydrolyze it, and we have to take. If you take directly, we don't have some sufficient enzymes to break this glycosidic linkage. So so you will get some stomach issues. Okay, but some animals, right, cow and other animals, they have some kind of enzymes. That enzyme can break this glycosidic linkage, beta glycosidic linkage. We have enzymes. That enzyme can break alpha. To break beta 1,4 linkage, you require some special enzymes. Those special enzymes are present in animals. Okay, other animals like cow and etc. But for human beings, we don't have those enzymes. So it is very difficult to break this glycoside linkage in human beings. That's why we should not take vegetables, right, like uncooked vegetable directly, right? For some vegetable we have to cook and we have to have it. Yeah, is it clear, student? Any doubt? Any doubt in this, students?
No, sir.
Yeah, good. So with this we have completed polysaccharides. Okay, so in syllabus we have only starch and cellulose, so remember all this information. It's very, very, very important. Okay, then any doubt, students, up to this? Any doubt?
No, sir.
Okay, so we'll have few questions, then we'll move further. These questions are there in the in-text questions in your book. It is given that glucose or sucrose are soluble in water. They are soluble, right? Freely soluble, you know very well. Glucose is freely soluble, sucrose also freely soluble in water. Whereas actually glucose consists of a 6-membered ring, right? Sucrose also consists of 6-membered ring and 5-membered ring. But if you take 6-membered ring compound called cyclohexane, this is called cyclohexane, right? Or 6-membered compound called benzene. These compounds, they are insoluble in water. Though they contain 6-membered ring, they are insoluble. Whereas glucose, you know very well, glucose also has 6-membered ring. It has 6-membered ring, but that is soluble. Why? What is the reason? The reason is that, what is the reason behind? Why glucose, sucrose are soluble in water? Because glucose, sucrose, they consist of many hydroxyl groups. Okay, see, in glucose we have hydroxyl groups. Let me draw the groups here. So let me show alpha hydroxy, OH below, here OH above, here OH below, here I'll put H, here I'll put CH2OH. So in this structure, see, I have 1, 2, 3, 4, 5 OH groups are there. 5 OH group. And in sucrose, I have already told you, we have 8 OH groups. 8 OH groups. Presence of these OH groups is very, very important. Why? Because they form, so glucose, they form intermolecular hydrogen bonding. They form intermolecular hydrogen bonding with water. They form intermolecular hydrogen bonding with water. This is the reason why, okay, glucose and sucrose are soluble in water, right? The basic idea is this: in glucose and sucrose we have many OH groups. OH groups are capable to form hydrogen bonding, so they can form hydrogen bond with water. Okay, if hydrogen bond exists between glucose and water molecule, we call these types of hydrogen bonding as intermolecular. Intermolecular means between two molecules. This intermolecular hydrogen bonding is responsible for the solubility of these compounds in water. Students, why these compounds are soluble in water? Because of intermolecular hydrogen bonding. These compounds form intermolecular hydrogen bonding with water, so they are readily soluble. Whereas cyclohexane, benzene, this is benzene, right? In this compound there is no hydroxy groups. They are non-polar compounds. Water is polar, so they are insoluble. They cannot form hydrogen bond. Okay, so they are perfectly insoluble in water. Okay, remember this. This is a very important one. This is given in the in-text question, right? Then the next question. What are the expected product of hydrolysis of lactose? Students, you take lactose, this is the question they are asking, lactose, and carry out hydrolysis. Yes, what will be the product? Anyone? What is the product of hydrolysis of lactose? Lactose is a disaccharide. Correct? What's the answer, students? Anyone? Hello? What's the answer, student?
Beta D galactose and beta D glucose.
That's all. Good. Good. So the answer is this. Lactose is a disaccharide which on complete hydrolysis, if you do hydrolysis, lactose, the glycosidic linkage will break and you will get beta D galactose plus beta D glucose. These two are formed. So it consists of two monosaccharide units, beta D galactose and beta D glucose. Very simple. Then the next question, how do you explain the absence of aldehyde group in the pentaacetate of D-glucose? See, question is this. You take pentaacetate of D-glucose. I have already told you pentaacetate of D-glucose, okay, pentaacetate of D-glucose does not contain free aldehyde group. Free aldehyde group is not there. How to prove this? Because it is in cyclic structure. Okay, D-glucose in cyclic structure, when it react with the acetic anhydride, form pentaacetate derivative. Okay, so let me show the structure, students. Pentaacetate derivative of D-glucose. This is the structure. 1, 2, 3, 4, 5. I'll take alpha form. So I'll put H here. In OH, remove the H and put COCH3. Acetate group. Acetate. Similarly here, above H, in OH, we'll put OCOCH3. Similarly here, hydrogen, and here we have OCOCH3. And here we have OCOCH3. Okay, and here we have CH2OCOCH3. Students, this is the pentaacetate of D-glucose. Okay, these are all called acetate unit. Acetate. Right, like this we have 5 acetate units. Now listen, student. In C1 carbon, that is anomeric carbon. That is the anomeric carbon, right? Anomeric carbon, there is no free OH group, no OH group. OH group is not there, see. Now the group present is acetate group, not OH. If OH group is there, then if I put this compound in basic medium, the ring get open up. You will get free aldehyde group, but there is no OH group now. There is no OH group. Therefore, the anomeric carbon does not contain OH group. Therefore, the ring structure, right? It is opening of ring structure is not possible here. Therefore, this group, this compound pentaacetate of D-glucose, they does not reduce. It does not reduce tolerance reagent. It does not reduce tolerance reagent. It does not reduce tolerance reagent. It does not reduce Fellings solution. Fellings reagent or Fellings solution. It does not reduce. It does not form oxime. It does not form oxime. Okay, these are all characteristic reactions of aldehyde group and keto group, right? Oxime will, keto group will also give oxime, but they are characteristic reactions of aldehyde. Since these tests, your pentaacetate does not show tolerance reagent test, or it does not reduce tolerance reagent, it does not reduce Fellings solution, it does not form oxime. Therefore, it clearly indicates that aldehyde group is not there. There is no free aldehyde group. So you can prove the absence of aldehyde group, okay, by using this test. You carry out tolerance reagent test, Felling's reagent test. Tolerance reagent consists of basic medium. Felling's reagent consists of basic medium. But in basic medium, you require OH group here. You require OH group to open the ring. But there is no OH group in the anomeric carbon. There is no OH group in the anomeric carbon, so ring opening is not possible here. Therefore, pentaacetate of D-glucose, okay, does not contain aldehyde group. Very important, keep this in mind. Okay, yeah, students, any doubt? Any doubt up to this?
No, sir.
Yeah, right. So with this, we have completed carbohydrate part, and we'll continue the amino acids and proteins in the next class. Okay, I stop here.
Thank you.